Well, howdy, folks. Welcome back to another episode of the podcast. Great question for us today. Where is this barrier located? Now, this is a comment/question on a video I made briefly describing the blood-brain barrier, what it is. So essentially, they're asking, where is the blood-brain barrier located? It covers all of what's referred to as the brain parenchyma, which is a fancy scientific word meaning like the brain tissue, so all the neurons, glial cells, etc. It covers pretty much all of it. Now, it's a fairly simple question, but the answer is quite involved. So I'm going to go into some explanation here, some nuance, and then hopefully provide a lot of value to you all. All right, as we are on this journey of trying to learn more. So what I'm going to do is I'm going to go over what the blood-brain barrier is, just in case people didn't see the previous video. Shame on you, but I'll link it anyways. Go into detail, and then I'll go into some nuance, in my opinion, on answering this question. So the blood-brain barrier. So the brain is full of blood vessels. There's tons of capillaries, etc., that run all throughout the brain. And the purpose of that is because the brain needs a constant supply of vital nutrients. A couple of examples of those vital nutrients are oxygen and glucose. Just hold your breath. You can't last very long. And then if somebody is without oxygen, in a very short amount of time, the brain starts to die. So it needs it all the time. All right. Food, we can last for a while without food, but oxygen, not very long. All right. So the issue runs with, you got to get these vital nutrients, but there's also potential pathogens that exist in the blood. There could be bacteria, fungus, viruses, drugs that exist in the blood. And so the brain being the most vital organ, because that's what controls the whole body, there needs to be protection from these harmful substances. So that's where the blood-brain barrier comes into play. So I'll put a picture up here. You have the capillaries, which is the small little blood vessels that are run all throughout the brain. And then the blood is flowing through what's called the lumen inside those capillaries, right, with those vital nutrients and also those pathogens. Well, lining those blood vessels, those capillaries, are endothelial cells. There's all lining up all throughout those capillaries. Now, in between those capillaries, you have a network of proteins. It's occludins. Claudins are the ones I remember. And those proteins form what's called tight junctions. And what the tight junctions do is it prevents paracellular transport. So it prevents those bacteria, fungus, etc., from going in between those endothelial cells. So you don't want viruses to go in between the endothelial cells then to get into the brain parenchyma, the fancy word again, the brain tissue. You don't want that to happen. So really, in order for substances to get into the brain, it's got to go through the endothelial cells. Now, if you have a molecule that is nonpolar, meaning that there's not really a difference in the charge, either positive or negative, from one side of the molecule to another side of the molecule. Usually a nonpolar molecule will be something that's fat soluble. Then it can use what's called passive diffusion. It'll just go right through its fat soluble. It'll go right through the lipid bilayer and into the cell and then come out the other side and then be able to get into the brain tissue. Well, for vital substances, for example, glucose, that are water soluble, so water is a polar molecule. I'll put a picture of it here. You have the hydrogens, which tend to be more positively charged, and then the oxygen, which is more negative charge, so it's polar, it's different charges. Well, if it's a polar molecule, it's normally water soluble, so think of water and oil. They don't mix, so the cell membrane is a lipid bilayer. So you're trying to get a water-soluble molecule through a fat membrane, not really going to go too well. All right, so something like glucose, which is water soluble, well, there are specific transporters, essentially like gateways, you could think, that allow the substances to get through the lipid bilayer without having to deal with getting through the lipids, without getting through the fats. All right. So glucose, there is a transporter called GLUT1. All right. And there's different. There's like GLUT1, GLUT2, GLUT3, GLUT4. Those are the ones I remember. But GLUT1, glucose transporter, it allows glucose to get in through those endothelial cells. And then it also allows it, I believe, to get into the astrocytes. I think it's GLUT3 that allows glucose to get into the neurons. All right. GLUT4, I believe, is what allows it to get into our muscles. And GLUT4 is insulin dependent. So that's where you get the whole idea. You need insulin in order for the glucose to get into the muscles. In the brain, it's independent. You do not need insulin in order to get the glucose into the brain through GLUT1 and GLUT3, if I remember right. So that's just an example of that. Not everything is blocked by the blood-brain barrier, as we described at the beginning. You need those vital substances to get in, but you don't want stuff that should not be getting into the brain to get through the brain. All right. So that's the endothelial cells and the tight junctions. Well, then there's what's called the basement membrane, the basal lamina. It goes basal, means it's facing towards the, on the side of where the brain is. Well, embedded in that basal lamina, the basement membrane, are pericytes. Right? And pericytes do have a number of functions, but they also help to uphold the actions of the blood-brain barrier. And pericytes can have phagocytotic and pinocytotic characteristics. Pinocytosis, meaning that essentially, like if you have like small solute particles, and then they get on the membrane of the pericyte, well, then that membrane will kind of indent in, and I'll put a little animation up here to describe it, indent in, and then the extracellular fluid along with those little solutes get into that little crevice that's created in the membrane, and then it pinches off at the top. And then in this case, the pericyte can then metabolize those solutes to help get rid of it. You know, essentially, if it's like bacteria or fungus or something that's in the extracellular fluid there. And there's also phagocytosis, where it actually eats. It can actually engulf the substance and then, you know, help to neutralize it and get rid of it. So the pericytes they're all very important, but pericytes are important in terms of getting rid of the foreign substances that may have gotten through the endothelial cells. Right? And then we get into the astrocytes. Right? And the astrocytes, I'll put a picture up here. I believe it's Latin for star cell. So you have the middle of it, you know, where the nucleus is, and then you have all of these arms that spread out it looks kind of like a star. But at the end of those arms, you have what's called the end feet. And those end feet will wrap around the endothelial cells and the basement membrane and the pericytes. And the astrocytes have a number of functions. It's really fascinating to read about what astrocytes can do. But astrocytes are the location where glutathione is produced. We talked about glutathione in a previous video. And that's important for detoxification of like mercury. If you get mercury into your brain and it gets into the endothelial cells, which we discussed earlier, then glutathione, which is produced in the astrocytes, then can help chelate the mercury and then efflux it, means remove it from the endothelial cells back into the blood, which we can then excrete it, because you don't want mercury in the brain, because that's a neurotoxin. You can cause neurodegeneration there. So astrocytes are also part of what's called the NVU, the neurovascular unit. So you have your presynaptic, postsynaptic neuron. Then you have an astrocyte here, which is normally called the tripartite synapse, is what it's called. We talked about that before. But they all communicate. The neurons, the astrocytes, the pericytes, the endothelial cells. There's this communication between all of it. It's called the neurovascular unit. And this is where all of them are beneficial in it, but the astrocytes come into play here as well. Because let's say right here. I'm pointing, for anybody just listening, they're not watching, I'm pointing like right at the top of my forehead. Like for whatever reason, right there, there's neurons that are firing, which is normally doesn't happen. My neurons don't really work that much. At least that's what people tell me. They say I have excrement for brains there, but oh well. I guess maybe I have neurons in the excrement. But let's just say right there, the neurons start firing a lot. Well, when the neurons are working a lot, what do they need? They need energy. They need oxygen. They need glucose, etc. So what will happen then is the capillaries then will dilate to allow more blood with more oxygen, more glucose, etc., to get to that area of the brain to supply it with the raw materials in order to work more. And conversely, if there's area of the brain where the neurons aren't working really that much, which for me is usually all throughout the brain, they don't really work at all, then they will constrict. You don't need as much. So it's this constant flux of where blood and nutrients are needed versus where they're not needed. So the astrocytes play a role in that. So that's like a brief description. I mean, boy, if you really wanted me to, and I could do a deep dive on the blood-brain barrier, I mean, that could be hours long podcast, all the stuff that it could do. And I certainly am up for the challenge of studying and trying to get all the information in. You know, it's difficult with excrement for brains here, but I think I can do it. But let me know and I'll get on that. It may take a while, but I'm up for the challenge. So that's kind of like an explanation of the blood-brain barrier. So again, it kind of covers pretty much all of the brain parenchyma there to protect it from pathogens. Now we get into the nuance of it. Well, there's also something called the blood CSF barrier, right? And that is the cerebral spinal fluid. Now, where that exists is like, if you go, stick a pin right into my brain, kind of like in the middle there, I'll put a picture up here, but you have ventricles in the brain, right? The ventricles are kind of like chambers of the brain, right? And in those ventricles, you will have something called the choroid plexus, and that's like a bundle of blood vessels that are there. And in that choroid plexus is where the blood CSF barrier exists. And so the function is similar to the blood-brain barrier, meaning that it allows vital substances, electrolytes, water, etc., to get into the brain because you need water. I believe that's through what's called aquaporins, or the membrane proteins that allow water, which is again a polar molecule, to get into the brain in this case. But it allows electrolytes, etc., to get into the brain. But it also will allow the efflux of harmful substances. I guess in this case, you could use the example of amyloid beta, which I'm sure if you've done any research on Alzheimer's disease, amyloid beta is a component of the amyloid plaque, which is what's seen in people with Alzheimer's disease. They have this buildup of this amyloid plaque, which amyloid beta is a normal byproduct of brain metabolism, but our body's got to get rid of that. So normally, I believe the amyloid beta is removed via the blood-brain barrier, but to a lesser degree, I believe it's also removed via the blood CSF barrier. So I'm using that as an example that the function, even though the structure of it is a little bit different, the function is similar in that it can allow vital nutrients in and then waste products to be removed. Well, the vital nutrients are brought in to make cerebral spinal fluid, which again can have several roles, but primarily it's protection of the brain from physical damage, right? It's also spinal, so it protects the spinal cord as well from physical damage. So let's say you get, hopefully not, but let's just say there's like a car wreck or something where you get like a whiplash. Your head rocks forward or back, and then there's a sudden stop, and then your brain keeps moving around. Well, the cerebral spinal fluid provides that liquid, that kind of like a cushioning, a shock absorption to help prevent damage or limit damage of that, mechanical, physical action there. at the cerebral spinal fluid, it will then go in through the ventricles, and then it gets into what's called the subarachnoid space, which is like the area between the brain and the skull. So it then encircles the brain. So again, it provides like shock absorption for the brain. So yeah, I just wanted to add that, to add a little bit of nuance, add a little bit more value to you all. Now, something else that I wanted to add in here that I'll finish up the episode with, I think you'll find really, really cool. In the fourth ventricle, I believe it's way down on the brainstem, right? There's an area of the brain that is not protected by the blood-brain barrier or the blood-CSF barrier. It's not protected, and it's called the area postrema. I'll put it up here because my spelling is horrible. Area postrema. And I like to think of it kind of like if you think of Middle Ages, back in medieval times, if you had the king, they would have the official taste tester, in case somebody wanted to poison the king, you would have the taste tester taste the food first, and if they were okay, they didn't die, then you have a pretty much assurance that the food is okay, it's not poisoned. So the area postrema is like the official blood tester of the brain. It will actually monitor the blood for toxins, etc. on that. Well, there's a toxin that pretty much almost all people consume that the brain does not like, and that is alcohol. So if there's a lot of alcohol in your blood and the area postrema can detect that alcohol, well, what does it do? It then signals to the stomach for the substances to go the other way, to pray to the porcelain god. Now, it's not the only reason why we vomit if you drink too much alcohol. Alcohol is very disruptive and abrasive to the stomach lining, so then that's another reason why. But that is one reason, is that your brain, that little area, the area postrema, will actually test the blood, and then if it detects that there's too much alcohol, it will then signal to the stomach to throw up. So I just thought that was interesting to add that little bit of nugget there for you all. But I hope that answers your question. Please keep them coming. I will get back to you as soon as possible. So please like, subscribe, share, all that good stuff. And until next time, much love to you all. Take care. Adios.